So in this experiment, we're going to be doing a Bradford assay to determine how much protein there is in our lysate. So first thing we're going to do is we're going to create a standard using PSA. We're going to load our lysate and add Bradford's reagent to figure out how much, take it to a spectrometer, and measure the absorbance. So first thing I'm going to do is label all my Eppendorf tubes with how much BSA I'm going to put depending on the concentration. And then I'm going to add the BSA to it, and then we'll add our lysate afterwards and then the reagents. So we're going to go from there. Next, we're going to load our lysate. We're going to add 10 microliters of our lysate to our solutions. BSA and all our proteins of the BSA and lysate, we're going to start adding our Bradford reagent. And it's going to be added to make our total solution volume of 600 microliters. So we're going to add 600 and go down as we go along. It's important to keep the Bradford reagent wrapped as it is light sensitive, so only remove the cap when you're ready to do this step. Two down. Because we're using precise amounts, it's important to count the number of notches and clicks as you work your way down your pipetter and make sure you're going down by the right amount. Now that we've gone to this part, we can do the 10 microliter BSA as well as our lysate, since they both contain the same volume of protein. So without changing the pipetter here. Final one. Now that we've finished, we're going to lightly vortex these just to make sure everything is combined. And then we're going to let these incubate for 5 minutes. Finally, our lysate. I'm going to close everything out. We're going to wait 5 minutes. Alright, so now the 5 minutes have passed and we're able to load our samples onto our 96 well plates. We're going to do so right now. So you're going to take your 96 well plate, and it's important to understand the order in which you're loading them. So we're going to load them from 0 to 20 and then load our lysate sample last. This is important when you go into the software so you can know what's the blank and what's not. So all you're going to do, open up your sample, and you're just going to load it like that. So I'm going to go top to bottom. And don't go to the full, don't push too hard on the second stop, otherwise you will create bubbles and that will impede the readings. Now that we're done with one of them, I'm just going to move it up so I know where I've done. And you're just going to work your way down the list here. Try to minimize bubbles as much as possible here. You should start to notice as you're going down in protein concentration, the color of your reagent is changing. It's becoming more and more blue, which indicates more and more protein is found. It's a good sign that everything is working as intended. And now that we've finished loading our sample buffers, with VSA for our control curve, calibration curve, we're going to add our lysate to the side, noting the position. Just going to add it to the left of the negative control. It doesn't matter where you add it, as long as you mark where you add it, you can recall when you put it into the software. Now that we've loaded the triplicates of both our calibration curve standards as well as our unknown lysate, we're going to cover with aluminum foil. It's best practice to cover with aluminum foil when you're not using it to make sure the results are accurate and because the UV lights don't mess with it. We're going to take this to our plate reader. We're going to look at the results.
Hello, today we'll be talking about the tutorial on how to use the microplate reader. So when you turn it on, it's going to look something like this. It's going to be the Epoch 2 microplate reader. So if it's not on, you're going to want to turn it on. So first, from the bottom, ensure that it's on from right there. And to turn the screen on, I'm going to click up there. Right, now that's turned on. We're going to use this software here, Gen 5. And you're going to wait for it to load. Right, now that it's loaded, you can either select a previously existing protocol or create your own. So we're going to use a new one. And when you click on a new one, you're going to get a screen something like this. And then you have different features. So in the first, you can select the size of the plate you want to read. In this case, we're going to pick a 96 will plate. Then you can have different tools. So you can first check the method of absorbance. So what is it reading? In this case, we're measuring absorbance. All right. If you click OK, you're able to select the wavelength of the absorbance you want, and up to 6 different absorbance wavelengths. So if you have different reagents you want to look at the same time, you're able to do so. In this case, we're going to keep it the same setting as 450, but do it depending on your protocol. I know your protocol says 595. Click OK, and that will lock that in. Next, you can set the temperature. So you click set temperature, and if it's a reaction that requires temperature sensitivity, you're able to set it from here. Next, you have shake. So if you want to mix your solution every once in a while, you're able to do so. Next, you can look at the kinetic studies. So we'll take the measurement at different time points. This is depending if you want a time series or if you just want the measurement as is. Also, you can have it monitor the wells at different intervals. Finally, you can have a delay, pause, start. So you have full control over your assessment and do your assay. Then you want to click OK when you're ready to go. And when it's done, the plate reader should pop out. Now, be careful about the plate reader. It's going to tell you, do you want to start using the plates? It's going to click OK when you're ready to go. So in this case, we're going to click OK. And we'll start taking measurements from the machine. Now, before we take measurements, you can also do something else. That's where you can set your plate parameters. So click here, you can set different types. So we have samples, you have your sample controls, and you have your blanks. So in this case, we're going to select samples and sample blanks. There we go. From this, just for as a demonstration. And here you're able to name your blanks and your samples, as well as assign them a color. So it doesn't matter what you name them. I'm going to leave them as the default name. I'm going to click next. Again, you can set the settings. I'm just going to click next and keep them as a default. And you're going to get to a plate like this. And this way, you can assign the plates and wells to different things. So for example, I'm going to assign these as the blank. You can just drag it. I'm going to assign the next 3 as the samples. That way you can stay organized, and when you download the file, it will be ready to go. Click OK. And that way, when you download the file, it will label them exactly how you want them, so you don't confuse them. Next, we take the plate, and we put it into the reader. It will say A1 here, so it'll tell you which orientation. Make sure not to flip it, otherwise it will cause a problem. And when you're ready to go, you just want to click OK. take the measurement. So you want to click read now. Select a new one in case there's an existing one. It's going to confirm if you want to take the measurement like we saw previously. Click OK. It will take it in and it will start reading. After it's finished reading, it will look something like this. You'll have to save your file. In this case, you can name it whatever you want. We're going to name it experiment1. And it's going to save it as a .expt file. I'm just going to click cancel. I'm not going to save it, but that's how you save it. And then you'd have the readout here. So if you want to take a picture, if you want to take it for your notes, you're able to do so directly from here. As we can see here, we're both blanks, so they have comparable readings. When you're done, take your well out, put it aside, and make sure you clean this well before you do put it in. So, and you also want to make sure it's not sticking out. So you see at the bottom here, you click the button once, and it will retract that. So when you're done, make sure it's retracted so it doesn't break by someone accidentally tripping it on it. Now that you have your measurements on the screen, you're able to take pictures of them and manipulate them however way you want, as well as save them and extract the files. And when you're done, you want to click X, save the files if you haven't saved them, get out to the epoch screen, and then turn off the reader, turn off the monitor if you're the last group from the day to make sure it's ready for the group tomorrow. And that's how you use the microplate reader.